We're going to do the unsupervised data mining general clustering now, and hopefully this will work right. Okay. So first, I'm just going to very high level just talk about data matrices, things we can do for them, and how we can think about clustering. So let's just, again, think about the typical situation in bioinformatics, very general. We make a table. That's what we do all the time. And so just for concreteness, let's think about a table where we have many different sites on the genome, okay? And we have many different things that can happen. These could be various transcription factors combined, or we could have different levels of expression if they're in different tissues and so forth. And, as we'll talk about, we can think of these as the points and these are the features, or we can think of it the other way around. But we have a big matrix that looks like this. And there's just essentially an infinite number of things you can do with these matrices to go blue in your face. And we're going to talk about, many of the methods. I just want to give you a sense. First, you can just do real simple stuff, overlap things in the matrix, look for enriched regions. We're going to talk about kind of simple clustering methods, and then we're going to spend a long time talking about spectral methods, and we'll kind of put that in relation to one type of clustering thing, and sort of SVD and a lot of its friends. And then we might spend, if we have time, talk about some of these more distributionally oriented things like LDA and so forth. And then later on, you'll hear about the deep learning. There's yet more methods for this that you'll hear, maybe these autoencoder methods. So first, let's just start simple things you can do with the matrix. In bioinformatics, all you do is look at matrices all day. And you think about things you can do. So the one simple thing you can do in your matrix is you can just look for, before you get into complicated clustering, and this, you can just look for various populated and depopulated regions of your matrix. You could have an unpopulated region and a highly populated region. And if you think about what's going on here, it's important to kind of keep the physical reality of going on. If these were sites in the genome, this would maybe be an area of the genome where there's a lot of activity, there's a lot of things being bound to, and this might be a genomic desert, nothing's happening, and so forth. And you could come up, I think it's kind of useful thinking about how could you kind of find significantly unpopulated regions. I'm not going to go into the detail of how you might do this. That you could imagine some sort of binomial type of thing, Poisson type of thing, where you would look at how likely you are to have nothing happening in a particular region relative to some expectation, then kind of call some regions that are significantly over or underrepresented, and then call those to be maybe deserts, or the converse being forests. Another type of calculation that people do very often in genomics, which it's actually a little less intuitive when you think of it from a machine learning perspective, but it's very common in genomics, is they do what's called aggregation analysis. So they'll have this genome, okay, so they have the sites on the genome here, and then they have the different features going down, and they'll pick some sites as anchors, okay? Let's concrete, what are they often? They're like the end of genes, the TSS, or maybe it's a binding site. And then what they'll do is they'll average the matrix, they'll recenter all the matrix around that. So they'll cut the matrix up, recenter around that site, you see what's going on here, and then they'll compute a distribution around that site. And this is often, for instance, they'll look at the chromatin signal around the TSS, or they'll look at the level of gene expression, across the gene and things like that. Very common type of analysis. This would be kind of aggregation analysis. Another kind of common analysis people do is they do saturation analysis. So they're often here, they might be thinking to themselves of, let's imagine that going down, we have genes, okay, going across, we have different sites in the genome or different genes. And going down, we have maybe different conditions that we're looking at gene expression in. And we want to see how many conditions do we have to look at till we've gotten everything kind of being expressed, or we've gotten complete coverage. And so they'll look at, oh, well, if they do one row, how much is covered? They'll do 2 rows, how much is covered? And then, of course, by the time they do 5 rows, all the genes are covered, or all the places in the genome are covered. Now, it's a little bit more complicated when you do these what's called saturation plots, because the order that you're going down is kind of arbitrary. So you could go 1, 2, 3, or you could go 1, 3, 2, and so forth. And so when you think about the ordering, or the which way you pick the first one, the first and the second, the first and the third, you'll realize that you have to kind of make a little like a box, a variation box here around each thing, depending on what order you traverse the assays in. And that it gives rise to this. And so this is a kind of very common saturation curve. And, people will do this often for, like I said, gene expression. They'll do it also for, have we, if we look at these 5 transcription factors, have we looked at all the possible things that could happen, or do we have to look at another 5 things to get a sense of all the different bases in the genome, what they're doing? And they're very common type of analysis. Now, another analysis that is very common, and first let's start with the simple way of doing it before we get more complicated, is just, sort of activity clustering or expression clustering. Very common type of analysis people do. So again, what happens here is we have our matrix. And the simple thing we're going to do is we're going to take our matrix, which remember we have, let's look at this one first here. We have our different, say, RNA experiments, okay, or different times. We have the different, say, genes across, right? And we're going to correlate each of these rows with each other. So we imagine we take each row and correlate it with each other, okay? And from that, we're going to get some sort of affinity matrix of all the different rows. So, if we have sort of 10 rows, there's a whole sort of 10 by 10 possible correlation matrix that we can get for this. And then, as we'll talk about, the infinite number of things we can do with this correlation matrix. We can PCA it, we can biplot it, we can go on and do things with it. But we'll talk about that in a second. Now, the key thing to realize is that you could also imagine doing this over columns of the matrix. So if the columns are sites on the genome, or let's say they're genes, you could, and imagine, just for simplicity, let's say this RNA is RNA in different tissue or RNA in different times. You can correlate all the genes. Are they on together? Are they going on and off in different times together? And again, you can make a big network of genes. Or now this, of course, could also be sites bound by different chromatin factors. You do the same thing with columns and rows. And as we'll talk about when we come to SVD, there's a kind of nice equivalency of columns and rows in these matrices. And let me just, final thing before we zoom into the abstract, let's just think a little concretely about this. So here's another representation of our matrix. This is just simply a signal profile of a particular gene. So here's a gene, which would be, for instance, a column in that matrix we looked at. And we have many rows here where we're looking at the expression of a gene. This, an early experiment. This is a classic gene expression experiment from David Botstein and Ron Davis. You can look at 2 different genes. This is from yeast. There's many possible relations. We can see, are they similar? Here's 2 that are very similar. These are 2 ribosomal proteins. These would cluster together very well. And then, of course, we might make a cluster. We might have a gene that clusters, but we don't know what it does. And we could maybe say guilt by association, it's in the same cluster. So this is the kind of simple-minded stuff you could do with these gene expression profiles. And what I think is important is we can take that matrix of all the gene expression profiles, and we can then find the things that really are similar to each other, and we can make a network. So we can make an affinity matrix, and then we can make a network. And this is just a picture of a network. This is, say, from yeast, looking at yeast gene expression. In yeast, there's about 6,000 genes. So there's about 18 million possible relationships between these two, between any 2 genes. 6,000 choose 2. And in these experiments, maybe of the 18 million, say about 500,000 of them are significant relationships. And so that's what you're seeing in this network. In network picture, you're seeing a piece of these, huge, matrix of 18 million entries, which ones are significant. Okay, so that's a kind of concrete thing. Now we're going to talk about a huge amount of machinery for dealing with these matrices and finding groups in them and so forth. And so we're going to start on now. So now let's just start on the sort of high level. There's a number of different ways of thinking about this clustering, these type of clustering approaches. This is a general stuff people do. You can read about in lots of statistics books. This is from this Scikit website that shows different things. So let's just go through some very high level things. So there's centroid-based methods. And here what we're going to do is we're going to essentially put down centers, and we have to pick the number of centers, and then we're going to find which data points fit with those centers. And so in an abstract way, we think of each center as kind of being in charge of what's called the Voronoi diagram. Those are the points that are closest to that center that are not closest to the other things. And a method that's very associated with this type of approach is k-means clustering. I just want to give you a sense of how k-means clustering works because it's such a common method. Here you put down a number of cluster centers. You find the data points that are closest to these centers. So for instance, all these x's are closest to number 1. All these x's are closest to number 2. Then you recompute your centroids. Maybe you find the center point of all these points in your cluster center moves, and then you redo the method again and so forth. And you sort of stop when the centroids stop moving. That's a sort of baby k-means approach. Now, one of the problems with k-means is you have to decide how many cluster centers you want ahead of time. And that's a big decision that you might not want to make. Okay, and then a sort of generalization of k-means type of approach is a kind of distribution-based methods. And here, instead of thinking of centers of clusters, you think of them as kind of like Gaussian. So maybe there's a Gaussian and it's a distribution, it has a standard deviation, and you're going to try to see which of these Gaussians your data points goes to. So people often talk about like Gaussian mixture models and so forth, and you could generalize this in all sorts of ways. We can see that the LDA model that we're going to talk about is kind of like one of these distribution type of approaches. Now, another thing you can do with all your data points, remember we talked about this earlier, you can just do hierarchical based clustering. So you're going to find the 2 points closest together, you're going to merge them together, you're going to find the distances to all the other points, and you're going to merge them, and you're going to make a tree or a dendrogram. Now, this is very common with sequences, but you can, of course, make one of these dendrograms or trees trivially with the expression data and the other types of data. And this provides a kind of hierarchical view of the relationships where, these A-B things, they could be gene expression profiles that are closest together than C-D and so forth. But the problem is to get clusters, you have to kind of pick a threshold. You might say, well, I'm going to cut the tree here, and then all the things that are together at that cutoff, they're grouped together. And, that's one type of issue that's, it's like picking the number of centers in k-means is a kind of arbitrary choice. And also, we talked about this when we talked about sequence clustering. There's other issues that come up. Are you going to demand single linkage, multiple linkage? That means when you're linking something to a cluster, is it going to be, does it have to link to everything or just one thing? There's an infinite number of decisions, but this is the type of thing that people often confront with agglomerative clustering or bottom-up type of approach. Let me tell you another broad category of methods is what's called density-based methods. And here you imagine your points here in a high dimensional space. And here what you're doing is it's almost like kind of edge detection. You're looking for very sparse regions in that thing to define boundaries, okay? And actually, this is a little bit like the MSB method, a mean shift method that we looked at for structural variance earlier, where you were kind of looking for the gradient and kind of going down to the base and center, and you were then getting a boundary between things. So another method for this, a very popular method from this is this DB scan method. Many people have different methods for this.